Welcome to the fifth seminar in our Bioentrepreneurship seminar series. Today we're talking about therapeutic drug development and regulatory approval, and we have the privilege of listening to Dr. Francis Richmond, who's the director of the International Center for Regulatory Science at USC. We have the support of Pasadena City College, CNSI, and the USC Michelson Center Bridge Institute. Before we get started, I'm going to give everyone a brief introduction to our work and our team at BCLA. We are 22 team members, and we've been working for about 6 years to generate now what we have is 6,000 subscribers, and we've partnered with over 100 organizations and institutes in and outside of LA. And a little shout out to our core sponsors, CNSI, at UCLA and USC Michelson Center Bridge Institute. Our mission is to inspire, educate, and connect emerging scientists and entrepreneurs to grow and diversify biotech in LA. And if you've been to our events before, you know we organize networking mixers, seminars and panels, symposia and summits, and career conferences. And if you haven't, and this is your first time, welcome. We partner with many organizations, both in industry and in academia. And today we are very excited to organize this event with our partners at LA Bioscience Hub. And I'm Pasadena City College, but now I'll hand over to Jordanos Gabritatios, who's the executive director of LA Bioscience Hub, to introduce our speaker.
Thank you, Sadhna. Welcome, everyone. Thank you for joining us for today's webinar. My name is Jordanos. I'm the Executive Director at LA Bioscience Hub. We are a geographically focused nonprofit focused on LA's East Side, and we work to create a diverse workforce that is ready to enter LA's growing bioscience sector here. This is our fifth summer doing the entrepreneurship seminars as part of Biotech Leaders Academy. We're so pleased to partner with BCLA to present this webinar series online and to make it more broadly available to a larger audience. Our purpose with this series is to introduce to you the most important components of entrepreneurship in the biotech field. We're extremely fortunate to have our guest speaker, Dr. Francis Richmond, here with us today. Before I turn it over to Dr. Richmond, just a little housekeeping. If you have any questions during the presentation, please type them in using the Q&A feature in your Zoom control panel. We will be taking questions throughout the presentation, so you don't have to save them for the end. Without further ado, I'll turn it over to you, Dr. Richmond.
Thank you. So let me just share my screen. So thank you very much for the opportunity to talk with you. I have to, before I start, say how impressed I am with the activities of the Biotech Leaders Academy. I think that the work that they do with young people from diverse backgrounds is something that's just terribly impressive, and it's my great privilege to be some very little part of it. I know, though, today we have a bigger audience than we usually do in the classroom. So what I'm going to do, though, is go over the world of product development and regulatory science in 30 minutes or 40 minutes, which is kind of a reach. So this will be fairly high level. Now, at some points, we'll stop and ask questions because I'm hoping that the questions themselves will be more useful to you than my talking. So please feel free. We'll try to stop 3 or 4 times so that you have an opportunity to ask questions. And if there's any time at the end, you can ask them then as well. My email is here, and I'm always happy to answer questions as well. So you've got a medical product, and today we're really honing in on drugs, although much of what I say will also be applicable to devices. I mean, the LA region is a huge device hub as well as a drug hub. So with any luck at all, this will advance. Now, whether you're developing a medical product or a drug, a device, whatever, cosmetic, you're just like any business. And so what do you have to do? Like any business, you have to make something, and then you have to sell it, right? What is the difference is that typically medical products take a lot longer to develop, and they're more expensive to develop. So some estimates for drugs have suggested that the drugs we're making these days are costing more than $1 billion to develop, and they will take anywhere from 5 to 20 years to come to the market. And some of that time is taken up in regulatory agencies that are trying to ensure that these products, which are dangerous if mismanaged because they're going into the human body, are very safe and also efficacious, so that you will not waste your money on something that doesn't work. Now, at the same time, when we think about selling a product, we also have to think about agencies like health technology assessment organizations that decide if not only if the product is okay to use, but is beneficial from a cost point of view, is reasonable. And in addition, we concern ourselves with formularies, that is, those lists that end sellers may have, insurance companies may have, that will allow you to be reimbursed for a drug if you need it. So when most people think about medical product development, they have an idea and they're thinking, this idea could be commercializable. What they perhaps are not so aware is that the likelihood that any single molecule that is discovered in a laboratory will reach the point where it can be sold on the market, that's very small. Here you see a picture of sort of 5,000 to 10,000 compounds going through drug discovery, preclinical, clinical trials, FDA review, and then into manufacturing. Some estimates place the fact that 5,000 compounds can turn into one FDA-approved drug. And there are a number of things that have to be shown on the path that we'll talk about today. So in some ways, this is just a bit of a high-level table of contents for things I want to touch on. Now, for the average person who is in a laboratory, and many of you are from a laboratory background, you understand a little bit about discovery. You've talked a little bit about that. You know a little bit about patents because of the previous talks that you've been given through this series. Then you don't know what happens. There's a certain sense that magic happens and suddenly you get a commercializable product. And what we're going to talk about today is this box called magic here that isn't really magic. What do we do along this path? And so I'm going to identify, 4 major hurdles. The first you've already talked about, and that's making sure that your intellectual property is secured before you go talking about it to everybody in the environment, because there are rules with patents that once disclosed in public, it makes it more difficult to secure patent protection. Then we have to talk about getting the product ready to go into clinical trials. Clinical trials are required for drug products to get to market. After we have that clinical evidence, we need to gain regulatory approval. And once we've got regulatory approval, we have to be sure that we have the mechanisms for reimbursement in place. So here's just a little sketch of what some of the principal requirements will be for you. If you've got a compound, generally there's a stage of animal testing, and we'll talk about that in a minute. We also have to remember that the product can't just be given as a raw chemical. It has to be made in some kind of formulation. Could be a pill, could be an injection, but smart scientists are going to have to work on the chemistry to make sure that the product will be absorbed and metabolized and excreted in a reasonable amount of time. We also at this point are looking at what the product is going to claim to do at the end. This is really important. Many people don't recognize that you have to focus your development activities, even as early as this, on what that product wants to look like? What is its profile? What's its target profile? So that we know what we're aiming for. We know how to manage the animal and the clinical trials to make sure that we have the evidence that we need to be persuasive about this. So once we've done this stage, we'll talk in a minute about that. We have to put into the FDA what is called an investigational new drug submission. Now here I've also put IDE, that's an investigational device exemption submission. They're sort of the same. They both are seeking the permission of the FDA to proceed with clinical trials. Now I've also shown a dragon because the submission here is not necessarily straightforward to the beginner. There are many, many elements that the FDA wants to see in certain ways. And so we have to know what those ways are in order to focus our animal testing, our formulation, and our claims. After we've got that permission, which is permission to go into clinical trials, we will go forward with a series of clinical trials. And again, dragons, because clinical trials have lots of challenges. Then if we finish these and we have persuasive evidence that the product works then and is safe, then we'll go forward to give a marketing submission, which we call a new drug application, or for devices, it would be a pre-market approval application for a device that was very challenging. I'm not going to talk much more about devices today. But the whole point of what we're going to do here is getting to yes. Many people feel that the regulatory stages are all about getting to know, but we're not trying to get to know. We're not trying to get in the way. We're trying to find the most efficient and effective path to follow to develop the product so that we don't waste time and so that we don't waste money doing things that we shouldn't ought to do. So again, what we're talking about is a few stages of drug development: a preclinical stage, an investigational new drug application, a clinical stage, a new drug application, a marketing stage, and remembering that any one of these stages, including after it's on the market, the product might come off the market because it shows late stage safety problems, or it's supplanted by another product that's even better. Okay, let's talk a little bit about the preclinical stage. Generally with a drug compound, as many of you know, a first stage is to expose it to some sort of cell model. We're trying to find out here primarily, is this drug going to show some evidence of working? Does it attach to a receptor appropriately? Does it look like it has a mechanism of action that will support a claim? After that, what we are going to do is a stage called pharmacology. Now, pharmacology is the stage when you're looking at, again, what I call efficacy. That is, does the drug do something useful? So we first test it in cells, but cells are still limited in their ability to tell us how a whole animal would react, how a whole human would react to this product. So we want to know not just what the drug does to the receptors that they target, but we want to know what that drug might do to other off-target organs or tissues. We want to start getting a sense of whether it has off-target effects or whether it will be an unsafe product for the liver, for the kidney, for any other part of the body. Once we have a sense that we have a lead compound that looks pretty good and offers advantages potentially over what's already on the market, we will advance to a series of studies that are called toxicology studies. Now, this is the other half of the agenda, right? One half is efficacy, the other half is safety. So we're going to look at the safety of the product in order that we can predict its toxic effects in man. So depending on the product, the product's nature will shape the testing that is done. But it is generally a rule that for most garden variety drugs, we will need to test that drug in 2 species, often in a rodent of some kind and in a larger animal, so that we can get an idea of how it interacts with different species. Sometimes a drug will be safe in one, but not so much in another. And of course, these species are not humans. So that doesn't mean that the way the animal reacts will necessarily be the way that the human reacts. So I like chocolate. I can eat chocolate quite nicely. But this dog cannot eat chocolate quite nicely. They have different enzyme systems, different body systems. So while the animal testing is very commonly quite predictive, we still have to keep in mind that people may be different. Nowadays, you see that we have a lot of new drugs on the market that are called biologics. And biologics are proteins. They typically aren't given by mouth. They're given by injection because proteins are digested in the stomach, so they don't get absorbed properly there. These have a slightly different toxicological approach. For example, we often there have to use non-human primates to do some testing. Think of the COVID vaccines as well. They are biologics. And oftentimes, one of the biggest safety factors with those kinds of products are allergenic responses. Typically, species are very different in their allergenic responses to proteins. So it is quite often the case that any protein that's going to be good in humans cannot be tested in most animals or even in any animals, simply because the animal will immediately mount an immune reaction and try to destroy the protein rather than showing you its true effect. So with normal garden variety drugs, synthetic drugs, we tend to understand them well enough to know what kinds of adverse events we might get. And they typically can have a lot of off-target effects in the body. With biologics, a little different. They don't tend to have so many off-target effects in other parts of the body, but they do tend to be allergenic, immunogenic. So, and that's a very serious problem. So again, we shape our animal testing a little different. Okay, let me just see if this will. After we have that information, we go to another, this is often done in, of course, in parallel, as I showed you earlier, but we ask ourselves now, how is this drug going to be handled in the body? How fast can I get it in? So if we're delivering a drug orally, we know there will be a lag. It'll take maybe half an hour to absorb that drug. For giving an injection, of course, that's not an issue. You got it immediately. In a sense, that's a safety issue. You can't get it back. But different drugs are absorbed or introduced into the body in different ways. And the way that we formulate it can affect how it is absorbed through the gut wall, for example. And that's a serious consideration. We want to make sure it is absorbed. Some drugs are hard to get past the gut wall. So our chemical experts have to help us to find ways to manipulate the drug in order that it can be absorbed more readily. Then once it's in the body, we want to know how fast it is broken down. A drug with a very short life, say it's metabolized in 15 minutes, that's probably not what we want. Similarly, we don't want a drug that's broken down, well, maybe we do, but some drugs we don't want broken down a month later, because we may need a short-term action and we don't want that drug hanging around and causing problems to the body. Now, in some cases, though, we will engineer the molecule to stay longer in the body. If we've got some kind of an ongoing chronic problem like osteoporosis, we might want a drug that we only need to inject once a month. So again, we need to understand our kinetics and we need to figure out how it's going to be managed. Now, how fast do I get rid of it? Some drugs are metabolized mostly by the liver, some drugs by the kidney. We need to know that because if we have patients who have liver dysfunction or kidney problems, we know that we have to be careful about dosing those patients, because if they have difficulty getting rid of the drug, it's going to build up more quickly in their system, and there's more likelihood that they will have adverse effects from that high level of drug. So we will do the kinetics in animals, but knowing that animals are a little different, when we get to human studies, we will also want to know that. We will want to test that. And of course, we are concerned about safety. We've talked about the fact we'll watch any animal that gets this product for its adverse reactions. Something animals don't tell you very well. You can't say to your animal, you have a headache. You don't know if the animal has a headache. You have to do very indirect tests or look at blood levels of maybe some compounds that suggest the animal isn't doing well. So we do have to do much safety testing in humans. Of course, in humans, we're also going to try to figure out if the efficacy that we saw in animals is translatable. So now we're starting to, of course, edge into the clinical trial phase. And here's what we have if we don't keep ourselves organized. You know, the whole point of the exercise of figuring out how to develop a strategy for drug development is making sure you're not left with this, which now will have to be organized and put into place. What you'd really like first pass is to have something that looks like this, that your testing is all neatly lined up and you're marching step by step as quickly and efficiently as possible to get that very important drug to the market. So we're going to have lots of approvals, right? We're going to need FDA approval, first at the clinical stage and then later at the commercial stage. When we go into clinical trials, we're going to need approval from something called an investigational review board. An investigational review board is a group of experts and lay people from the community who are associated with the particular site where you're going to do the human testing. They are going to look at your protocol, look at your forms that you're going to give the patient, including a form to get their consent. They're essentially the local overseer of your trial, and FDA is the distant overseer of your trial. There's another thing that people often don't know, and that is that before you put a product into human in California, you need to have a state manufacturing license. That can be quite a hurdle because you're still at a very early stage of your drug. You don't have your full-scale manufacturing in place. You may not have your documents, you know, all the documents for your different steps, your batch records, the other things are not very well developed. So this license really deals with a system called, you know, sort of modified good manufacturing practices, but you still have to have manufacturing that is sufficiently well documented and sufficiently understood and sufficiently consistent in order that that product can go into humans, because you don't want a product going into humans that is manufactured in an inconsistent way that is, you know, vulnerable to having maybe bacteria in it in case of an injectable product. Lots of considerations there. And we're not going to talk a lot about that here, but that's a whole nother subject.
Dr. Richmond, I have two questions in the queue. I think they're related to the slide with the puzzle pieces. So one of the first one is what % of drugs which have been granted permission to proceed to clinical trials actually get through to NDA?
Right, and those numbers are known. So at the phase 1, I may even have slides later on this, but at the phase 1 stage, you may get a drop off of, you know, 40%, depends on the drug. But typically by the time you move through that path, a good 50% of drugs or more will fall off. And again, a lot depends on the nature of the drug. I mean, some drugs we know very well, so we wouldn't take that drug to, that kind of a drug to clinical trials unless we were fairly convinced of its success. Others, not so sure. So if you've got something that's very novel, it can be very vulnerable at that stage.
The second question is this process similar for building business-to-business products, like natural product discoveries/development or genetically modified animals developed for lab use? I'm assuming by this process he means the clinical trial process.
Right. I mean, animals for research use, that's a very different thing. FDA governs products that are, first of all, products, and second, medical products. So they have to treat a disease or a dysfunction. So by that criterion, animals for research wouldn't fall into either of those categories. And they might be governed by other regulations like ALARP, but not by FDA. Natural products are governed by FDA, but under a different category. They're considered, if they're called dietary supplements, that is, which many natural products are, they're a special form of food and they have their own regulations. Natural products that are coming in to treat a disease will be handled, though, in this same way.
Dr. Richmond, the next question I think is very specific. I think it has to do with the California state license that you were mentioning.
Mm-hmm.
The question is, what do manufacturing licenses look like when dealing with biologics that have large batch-to-batch differences?
Right. And again, that would be something that you would want to, I mean, obviously, even our marketed biologics sometimes have batch-to-batch differences. It's part of the game of biologics. How large they are will be important. So you will have certain specifications for your product and you will have certain tolerances around that specification. So what you have to do is have a manufacturing capability that meets your specification. Now, sometimes at clinical trials, the margin around a certain specification can be fairly large, but you do have to make that margin safe, and you do have to make sure that your product that's made batch to batch fits in with that specification. Now, sometimes at the very earliest stages of clinical trials, you only have a singular batch, and that batch needs to be, again, meeting some specification. So in some cases, it's very finicky. You do not want, say, when the COVID vaccine comes out, that it's contaminated with another virus or with a bacterium. So that might be a very tight specification. On the other hand, you may find that the amount of, of protein that's in that ultimate product, that final product, might be variable, especially at early stages when you don't have full control over manufacturing. And I think that the California State Board does understand that. So they're looking for risks, whether your product poses unreasonable risks because it's not manufactured appropriately. That's for that reason, I think a lot of early stage developers will engage the services of a specialty manufacturing facility that has some form of good manufacturing practices in place and that can easily gain this kind of a manufacturing license.
Thank you, Dr. Richmond. There's another question here, but I think we might save this one because I suspect you're going to be covering that.
Okay.
Yes.
Okay. So as I said, the first stage before clinical trials is securing an investigational new drug application. It's a pretty serious document. It can have several volumes of information about the chemistry, as we talked about the production, thank you for the question, and about the animal testing. It also has your protocol that you're going to use. And so the FDA will look very, very carefully at this protocol, and it will have all the ways you're going to protect the subjects, including the informed consent forms. The IRB will also look at this information, but in a much less detailed way. So we're going to then send it to the Investigational Review Board as well. And as I said, they're associated with the site. And their main function is protecting the rights of subjects. So they're really going to be most concerned with the risks that this product will pose and whether that, the benefits are there that would justify such a trial. Okay. Most drugs, unless they're follow-on drugs or drugs for a new application of an old drug, will start with clinical trials in phase 1. Phase 1 typically used to ask, does this drug have obvious toxicity in normal healthy males? And that's probably what you took in school. Nowadays, males are not the only ones used for all testing. In some cases, they are because of the cycling of female hormones, and also because of the possibility of pregnancy. So we do not give drugs that are very untested to women who might be pregnant or might become pregnant in the near future. So for that reason, traditionally, you will see phase 1 trials often done on male subjects. And typically, the male subjects are normal, relatively young and healthy individuals. We can go in for a long time about how that happens. If we have a drug like an oncologic drug, a drug to treat cancer, that has a known toxicity, but we're willing to deal with that toxicity if it's a really important drug to save a life, then it is unethical to test that drug on normal subjects, obviously. So in that case, we will do the first testing in patients. In phase 2, we will advance a drug that shows no immediate or great toxicity to patients who have the disease. And in phase 2, you generally choose a group of patients, maybe about 200 in your typical synthetic drug pathway. You select them carefully so they don't have a lot of other conditions that would obscure, that would confound the results. So we want relatively few complications because of other things that are happening to them. And these patients are heavily monitored. We're looking very, very carefully at a variety of things so that we can understand as well as we can, first of all, whether the product is safe for these patients. Second, what dose should we use? So generally in these trials, we will give more than one dose, and we will try to understand exactly which dose is appropriate to go forward with. And we'll want to look at the first evidence that the drug is working. After we've done those patients, we want to advance to what we call phase 3 trials. Now we have lots of participants who have the disease state we're trying to treat, but they may have lots of other problems. They will also have very major differences, perhaps in lifestyle, in what they eat, in what they do during the day. And so that variability will affect the efficacy of drugs. Drugs are affected by what else you eat at the same time as you take the drug. They're affected by your habits, smoking, drinking, other things like that are very influential. And in addition, many individuals have specific genetic makeup that makes them more or less vulnerable to certain kinds of adverse events. So for example, you may be one of those people who reacts very badly to the drug codeine. Most people, we know the dosage that works well for them and they have very little problem with it. But we have a population of people who do not metabolize codeine as quickly as others. And so there's a tendency for the codeine to build up to higher levels in their body, and they may react very badly to the drug. Other people are what we call super metabolizers. They take codeine, it is out of their system very rapidly, then they worry, why is it that I don't even get an effect? So people vary in their genetic makeup, and we need to start understanding that. So phase 3 could have, and typically does have, more than one study. Going back to biologics, we tend to test biologics at this phase rather differently. If the biologic is very, very effective, and some of those drugs are very, very effective, we don't need so many subjects. We certainly don't need 10,000 subjects. And if these drugs are life-saving for a disease that has no other treatment, we may find that we can commercialize that drug through pathways like what they call the breakthrough pathway. We may find that we can commercialize that drug with as few as a couple of hundred subjects. So they can move very quickly to market through this phase. That can be quite surprising and stressful for the people who are doing the chemistry, formulation, and manufacturing scale up, because they are more accustomed to having a very long clinical trial phase and not having to sort of make sure that everything in the back end to produce the product is moving as quickly as the clinical trials might. So clinical trials are expensive. So just an old but clean slide, and you can see that it used to be that the preclinical costs were the major component, but about 20 years that started to switch, and we're seeing a growth of the clinical trial costs. So for new developers, it's very important to understand these numbers. Of course, they vary product to product, but it's very important to understand these numbers, because as you get into clinical trials, this cannot be supported by grant funds anymore, typically. You have to have a partner, and you have to be positioned to have the money to do this work. So you and the FDA are going to answer one question. All of these studies go into what we call a benefit risk assessment. So if the benefit from the drug is very large, then the risks that you might take are also larger. So if this drug is going to save your life and there's no other treatment, you'll be willing to put up with hair loss or some other side effect in order to get the effect. Now, if this drug is to treat something like, eczema or, upset stomach, you are not going to tolerate losing your hair. So we have to match the benefits and the risks. But in all of this, we have to understand, were the studies appropriately designed? Do they tell us what we need to know? Did they prove that the drug was efficacious and safe, and that the manufacturing facility can make the drug reliably? So those are sort of the three main questions that all of this is trying to do. We put that together in a very large submission, and we give that submission to the FDA. The FDA will typically come back to us, if it's a priority drug, within 6 months. And with a more traditional drug that perhaps is not directed at something that is very life-threatening, then we will anticipate some response within 10 to 12 months. During this period, you are not strangers. The FDA will be asking questions, seeking more information. Every time they ask you for something, the clock stops until you provide it. So do not believe that because we publish these numbers, they are real. It is not unusual for those clock stops to result in pushing an approval out 2-3 years. we try not to let that happen by making sure that as we went through the development path, we did everything that needs to be done, so there's no reason to stop and do another trial. The FDA is also very receptive to meetings. They're formal meetings, they're fussy to set up, and they take time and effort, but that can help to keep you on the right path. And hopefully, what you get at that point is an approval. Now, maybe I want to stop here for just a minute because I'm going to switch gears a little bit and see if there are any questions about this path.
There are. The first question that we've had for a while here is from Harvey. He says, very informative and well-organized presentation. His question is, what criteria are necessary to fast-track a drug through the FDA, and what parts of the approval process can be sped up?
Okay, that's a big question. FDA has 4 pathways, and they mostly all depend on similar things. So, first of all, the FDA needs to be convinced that you are treating a serious disease, a disease that could be life-threatening or cause significant disability, and for which there aren't already good therapies. So that's the first thing, is they're not going to do it for something that is not life-changing. Then each of those pathways has a different thing that they'll do. Of course, the priority pathway, I talked about shortening the review time. Other pathways will allow measuring efficacy on the basis of what we call a surrogate marker. So instead of waiting for, in some cases for you to die and to see if it shortens the time to death, they might look at something that is a marker. So they might look at, some form of cardiovascular dysfunction over time, or some kind of marker in your blood. So that's a little dangerous. So we know that, for example, cholesterol tends to be correlated with some disease states, but it isn't predictive necessarily. So surrogate markers tend to be used for those products that are quite life-changing. So yeah, being able to, but all of the things that they have really typically advance the clinical stage more quickly. So I think the focus is on the clinical stage there, not so much on the animal studies or the formulation, which still, of course, have to be reasonable. They have to be appropriate because they can be a source of of unsafe product. Not sure if that answers enough.
We'll go ahead to our next question, which is, how is this process different for lab blood tests?
Those are very different from this process for 2 reasons. One is they're considered to be devices, so they would follow a device path rather than a drug path. The other is that you're not exposing the human subject to the product itself. You're taking samples from the human subject, taking it to a machine, and then doing the testing typically. If the human is not getting the product, then this is tested in a very different way, and there are a lot fewer rules for that part of it. It's also typically within the realm of clinical laboratory products, and they tend to be handled not by the FDA, but by a different organization. So we call them CLIA products or CLIA lab products in some cases. And that's for diagnostics that need to be highly validated. So they would follow a different path.
Our next question is, why has the clinical cost increased dramatically over the years? What are the main contributors to this rise in clinical cost?
Right. There are a few factors that are very important. The first is the tendency to need more and more data. So the number of subjects has gone up. Again, some drugs are tested on 10,000 patients. Not just because you need more subjects, but you need specialized subjects, like subjects with kidney dysfunction or subjects who are, children. So those are adding to the cost, of course. The more people that have to be enrolled, the more expensive it is. It's also the case there are a lot of drugs being tested, and the number of tests on the individuals that are in the trial have gone up. So each individual person in the trial is going to cost more because there are many more tests. If we add an MRI to the protocol, that's a significant addition of cost. But there's another thing to think about. Many of these products, we want to enroll as quickly as possible. So we can't get all those patients very quickly in the US. There may be other drugs for the same disease state. It may be difficult to enroll the patients. So more and more, we have global trials. Those global trials allow us to find additional patients, especially patients who are treatment naive. We often want patients who haven't been treated with several other drugs up till now. So those might be recruited overseas. That adds to the costs. Even though perhaps the local costs are lower, the costs of managing the trial go up. So that adds, and also we're marketing globally. So we're trying to make these clinical trials also meet the needs of other countries. And some of the other countries require that the clinical trials have a certain number of their residents, of their citizens enrolled. So for example, China requires that at least 200 individuals come from China if they're going to approve that product in China. So all of those factors contribute to that growth of expenditure.
Dr. Richmond, thank you so much for taking those questions. You answered them beautifully. We'll save the remaining questions for the end and let you proceed. We have 12 minutes to the end, and I want to hear what else you have to say. Thank you.
Yeah, and again, we can stop at any point, but I'll move quickly through this because it's not the main thing. But we really do have to ask very early in our trials, who is my customer? So what are we aiming for with all of this? We want to know if it has better performance than the other products on the market. That's all up here in efficacy. We worry a little bit about a drug that would have worse performance. But equally, we also have to think about cost. And so obviously the thing we'd like most is something that costs less and has better performance. But oftentimes we find ourselves in this quadrant. I cost more and have better performance. Does that mean a drug isn't marketable? Of course not. We know that there are drugs on the market that cost quite a lot of money, but they are so useful that insurance companies are willing to cover their costs. But if we're going to get support, we have to remember that FDA approval is not enough. If we get FDA approval, that's no guarantee that this is going to be successful in the marketplace. So now we have more submissions, and submissions that are just as hard, maybe harder, than submissions to the FDA. So if we want to get supported by the government programs, we have to put in a submission to the Center for Medicare and Medicaid Services, CMS. If we want to have our product on the hospital formularies, we have to have material that can go to the formulary committees. And in certain countries, like Britain, everybody perhaps knows this, the product is first scrutinized by something called a health technology organization. In NICE is the acronym for the one in Europe, in Great Britain, and people say NICE isn't always nice. And they take about 2 to 3 years to assess a product. So if you've got a new product, it can spend considerable time in the assessment. Unfortunately, for many products that are not, priority products, that assessment doesn't start until the regulatory approval is in place. So what does that mean? It means you may have a couple of years where there isn't a clear path to insurance coverage, and you have to work with the insurers hat in hand to beg them to support your product until a main decision is made. And they ask a very different question. So, you know, I often talk to my students about Viagra. The center, of course, it's an approved product, it's safe, it's efficacious. But CMS would ask, is it reasonable and necessary for the diagnosis or treatment of illness or injury? And so first of all, is it necessary? So if it's a lifestyle drug, this bar is a lot higher. If it's treating something that otherwise is going to be difficult to treat, then the bar is lower. So with Viagra, it could be that insurance companies would be happy to approve it in a situation where perhaps there was a difficulty for, you know, a husband and wife to have a baby, and the alternative was in vitro fertilization. In vitro fertilization is much more difficult, much more expensive than taking a pill. So that might be a very specialized opportunity to have reimbursement, but you might not have reimbursement for a drug like this for other applications. The other key word though here is reasonable, and that has to do with the cost of the product. So they're going to be very cost sensitive. You may find though, for many drugs, it already fits into a benefits category, especially if it's a generic drug or a follow-on drug. That kind of drug may be covered already, but the amount that's being reimbursed is too low for you. And so you have to go to them and try to convince them that a higher reimbursement rate is important. And it may, as I just sort of alluded to, it may only cover certain types of people or disease states. So all of these things are difficult, and particularly in the US, where the insurance system is highly fractionated. So much money and effort needs to go into getting these products into a position where they can be supported by the insurance company rather than out of pocket. All right, somehow this is unhappy. Let's just go to the next slide. So what does this mean? I mean, at the end of the day, this means that due diligence is very complicated. So you have to make sure as soon as you can. We often say fail early, fail often, because this is such an expensive path. It's really important as quickly as possible to make sure that the product is good enough. And also to make sure that the development team is good enough. Because again, it can be very costly to get bad advice or to slow down because you aren't quite sure what you're doing. Are there safety issues? These are critical. A very important thing to think about is the path sufficiently capitalized? Do you have enough money to do what you need to do? Also, is reimbursement possible? Are there other drugs out there? Are they being reimbursed? Are you going to have a difficult competitive landscape? Especially remembering if you've got something that is very important to the market, your competitors are going to want to get into that market as well, and they may be also developing other products alongside yours. So essentially, I think that's probably a good place to stop, and I can leave the sharing up in case anybody wants to go back to a slide, or I can stop sharing. Up to you guys.
We have two more questions. First question, is this process the same for psychopharmacological trials?
Depends if the product is a new product or an old product. So if you've got something where you're taking a drug and you're just trying to understand better how it works for the claim it's already making. So then that's easier. Typically the FDA has a much easier job because the drug is already known, the safety is already known, the efficacy is already known. It's probably already produced commercially. And so that's may not even have to be done typically by the FDA and can be handled locally by the IRB unless you're introducing some new risk. Now, that's not the case if you're trying to expand its indications for something new. Then the FDA wants to know about the clinical trial because it may introduce new risks. But I'm not sure that, you know, those kinds of drugs are any different than other drugs, although you may have something else in mind and I'm not answering the question correctly.
We have one final question. Should your GLP tox batch be exactly similar to GMP batch?
It can be hard to do that. And so typically it is not, simply because oftentimes that GLP batch is made so early that you haven't got the equipment and other things in place that you will use for your GMP system. So what you have to do though is make sure that the GLP batch is sufficiently consistent with your desired specifications. And so you're going to want to make sure that clearly the product's identity is clear. It lacks contaminants or other things in the mix, that it is what you say it is. So there'll be a lot of testing involved. But it's very difficult to make a GLP batch under exactly GMP conditions. And you know, it's a big discussion about how close it needs to be. Certainly, it can't just be a casual product off the bench that isn't tested and validated, because then you would, you would never be sure that the animal materials were the same as the human materials. So they should share the same specifications to the extent possible.
Another attendee is asking, what is the difference between IRB and FDA approval?
The IRB approval is a local approval, and it only extends to the site the study's being done at. The FDA approval will extend to all of the sites that it's being done at. The FDA is looking at it through the eyes of eventual commercialization. The IRB doesn't care about that. It cares about the safety of the patients in the study. So the focus is just a little bit different. Because the FDA sees this as a potentially commercializable product, then they're going to be very concerned that you're doing the right things to get to market, whereas the IRB will not look at the rest of the path very much. They'll look at the animal trials to see if there's predictive information for safety. They'll look at the physicians who are doing the trial and the team a little bit, and the informed consent, but they often don't have the depth of specialists to really understand the protocol very well, to understand where the results are really going to be what you want directed in the right place. So they're different. The FDA submission is more heavily scrutinized and is typically much longer.
Okay, thank you so much, Dr. Richmond. I unfortunately have to call the end of this seminar. I want to thank you for sharing with us your wealth of knowledge, and I want to encourage anyone in the audience who still has questions, which we're seeing in the chat, to do reach out to you and ask you if that's all right with you.
No problem.
I'll hand over to Jordanos, and she's going to close us out today.
Yeah. Thank you, Dr. Richmond. We really appreciate you being here and sharing your insights, and thank you to everyone for joining us today. Just one final plug for our last seminar, which is next week, Friday, August 7th at 9 a.m., and the topic will be intellectual property protection strategies. So with that, we'll let everybody go on with the rest of their day. Thank you so much.
Thank you.
Thank you.
Bye.
Bye-bye, everybody.